Plant growth regulators or PGRs include auxin, gibberellin, cytokinin, ethylene, and abscisic acid. Auxin is the generic name for all natural and synthetic compounds with definite growth-regulating features. Naturally occurring auxins include indole acetic acid or IAA and indole butyric acid or IBA, produced at the apex of plant stems and roots. Synthetic auxins include naphthalene acetic acid or NAA and 2,4-dichlorophenoxyacetic or 2,4-D. Auxins are used in plant propagation as they cause rooting in stem cuttings. They also induce flowering, as in pineapples, prevent falling of young leaves and fruit, and promote abscission of mature leaves and fruit. Auxins are used in parthenocarpy, that is, the formation of seedless fruit such as seedless tomatoes from unfertilized flowers. They exhibit apical dominance in higher plants, that is, they inhibit the growth of lateral buds. This is why the shoot tips of tea plants are cut or decapitated to initiate the growth of lateral buds. Synthetic auxin 2,4-D is used as a herbicide. It uses the physical difference between the leaves of monocots and dicots to selectively kill dicots. Hence, it is widely used in agriculture and horticulture. Gibberellins or gibberellic acids or GA are extracted from fungi and higher plants. They are referred to as GA1, GA2, GA3, etc. in the order of discovery, and they are acidic in nature. GA3 was the first of all the gibberellins to be structurally discovered. Gibberellins have varied physiological effects on plants, such as an increase in the length of the plant body axis. This is why they are used in the lengthening of grape stalks and sugarcane stems to increase the yield. Gibberellins also improve the fruit's shape by causing elongation as in apples. They delay senescence or aging of fruit, and are hence used commercially to extend the market period by retaining fruit on trees longer. However, they also hasten the maturity of seeds, and hence are used to obtain early seed production from juvenile conifers. Commercially, GA3 is also used in the brewing industry to increase the enzymatic activity inside grains during the malting process to achieve even germination in all seeds in commercial beer production. In cabbage, gibberellins promote bolting, that is, lengthening of internodes before flowering. Growth promoter cytokinin plays an important role in the cytokinesis or the division of cytoplasm in a plant cell. They were first discovered in the form of kinetin, a purine derivative. One type of cytokinin, zeatin, was later isolated from corn kernel. And much later, naturally occurring cytokinins were identified in specific regions of plants such as the root apex, shoot apex, and fruit to promote growth. They promote the growth of lateral and adventitious shoots by preventing apical dominance. They also delay senescence or the aging of leaves. Another growth promoter, ethylene, is present in plants in gaseous form. It is synthesized in the organs of plants that undergo senescence, such as ripened fruit. Commercially, it is available in aqueous form, ethaphon, which can be easily absorbed by plants. Ethylene gas is released from the absorbed ethaphon, which accelerates the ripening of fruit by enhancing the rate of respiration and is called respiratory climactic. This property is used in the artificial ripening of tomatoes and apples. It promotes the abscission of leaves and flowers, and thus favors thinning of cotton, walnut, and cherry. It also promotes the apical hook formation in dicot seeds to protect the apical part of the shoot that emerges during germination. It breaks seed and bud dormancy and is hence used to enhance the germination of peanut seeds and potato tubers. It also aids in the lengthening of internodes in rice plants, thus protecting the shoot from getting submerged in water. It also promotes root and root hair formation, thus helping the plants increase their absorption surface. It initiates flowering in some plants such as mango, pineapple, and cucumber, thus increasing their yield. Abscisic acid is a plant growth inhibitor that regulates abscission and dormancy in plants. It helps plants cope with stress and is hence known as a stress hormone. In the case of unfavorable environmental conditions, it inhibits germination of seeds. In the case of drought conditions, it closes the stomata, thus helping the plant withstand the stress caused by lack of water. In fact, the function of plant growth regulator abscisic acid is opposite to that of gibberellins.